Got it. Let me get out of this for a moment. I need to get rid of the.
Good afternoon, everyone. Thank you for joining us. We'll go ahead and give it a couple minutes for individuals to file in, and we'll get started momentarily.
Okay. Or glitch with Zoom.
Okay, we'll go ahead and get started. Welcome to those of us who have just joined. This is the next member hour in our Circular Bioeconomy Systems series entitled Nutrition for Brain, Body, and Wellness. My name is Sarah Rodriguez, and I'm the host today. ASABE member hours were started as an opportunity for our members to learn, connect, and grow. In a few minutes, we'll get started, but first, just a few quick session ground rules. Please do keep your audio on mute throughout the presentation. If you have any questions, you can either type them into the chat, and we'll read them after the presentation, or after the presentation is completed, you can unmute yourself and verbally ask your question. This meeting will be recorded and will be available for later viewing on the ASABE website. Today, we're hearing from Charlie Messina, a professor in the Department of Horticultural Sciences at the University of Florida. There, he conducts a teaching and research program focused on harmonizing crop improvement and AI efforts for agricultural systems to generate the environment while providing nutrition security, improve human health, and adapt to climate change. During this talk, Charlie will offer a perspective to approach this problem by increasing the availability, access, and adoption of healthier foods. All yours, Charlie.
Well, thank you very much, Sarah. Thank you for everybody for joining. I'll start a bit different. Thinking about taking aim at current syndemic. Think about the syndemic as an interconnected and global scale problem. And this syndemic essentially stems from these feedback loops between agriculture affecting climate change and climate change having negative impacts on agriculture and human health, reduce productivity. And what we do, we keep fueling more inputs and lowering the health of our planet as we go through this vicious cycle. And it's certainly not a problem we can solve in one place, but certainly is something that will require rethinking how we approach this major problem. And at the center, we certainly find diets that connects to improve the human health. That's our objectives, and the tools, given my background, and not necessarily is the only lens that we can use, but is a lens I know how to use, is breeding. But so we can think about how we can engineer crops in order to reimagine agriculture. So is part of the solution to climate change, and how we can use breeding to improve the nutritional contents of our foods to improve the human health. And hopefully by disrupting some of these feedbacks, we can have a good go at reducing this problem at a global scale. So said that, I think a number is useful to bring awareness of the magnitude of this problem. This is a figure that was estimated by Rockefeller Foundation. The price tag just for the United States is $2.1 trillion a year. It's useful to get this magnitude and put in context of the investment in recent research and programs that are in place to adapt or fight this endemic. When you unpack this figure, we see that it's about half of the problem is related to human health, part is food insecurity, pollution, and non-reportable diseases, and some of the half of that problem is about greenhouse gas emissions, soil erosion, land use, and so on. So agriculture is generating about $1 trillion worth of value to society, but at the same time is creating twice as many problems as it generates. So the question is, how did we got here? And at some point, we were not that sophisticated, so we were hunter-gatherers. There were small communities of high collective intelligence, but in small groups, and agriculture introduced the first revolution. So what agriculture led us is, if you will, was part of the problem at the very beginning, is to enable society and these groups and this collective intelligence to grow up to a point that now we have major cities producing a major intellectual output, but is demanding a whole different way of thinking about food chains and food waste and how we can circularize the bioeconomy in terms to delivering the food and nutrition that is required in a whole different set of arrangements. So the problem that agriculture has today is that society, whether it's how it's organized and the social demands, societal demands, are changing faster than agriculture. We are requiring more and better nutrition rather than calories. We would like to have an agriculture with lower footprints, and we want to focus on the human and the planet health and not just production of calories. So that World War II post agriculture, that again, agriculture provide another solution, introduce another revolution, got us out of that trouble, is overdue to set a new transformation, so we can deliver on the ambitions of society and the demands. So that lead us to my breakthrough question, and certainly it's not the only one, but is centered on this crop improvement. So how can harmonize the current and the future crop improvement efforts? This is creating improved varieties, if we will, and artificial intelligence, so we are smarter on how we create these varieties and faster for more nutrition. So these agricultural systems can produce more with less, so less water can regenerate aquifers, for example, by providing nutrition. If we put selection pressure on nutrition rather than calories, we can improve the human health, and certainly we can adapt to climate change. I'm not going to answer that question alone. So this is not just a team that will do it. This is a team of teams and perhaps of teams. And the first one I would like to bring to attention is this Center of Internet of Things. This is an NSF center, which is about delivering solutions in terms of sensors which are biodegradable batteries, essentially enable us to monitor the state of the system, and more fundamentally will enable us changing the mindset from, for example, manage nutrients today based on a prediction using this quadratic plateau response or ill response to nitrogen, to manage the complexity, so we can, instead of predicting the unpredictable, nobody knows what the weather is going to be like, we can monitor and implement control systems that is going to adapt in real time. So with the technology can help and enable us by quantifying what is the impact that we are causing on the environment and take corrective actions. That's one one example, so we can start reducing the problem of contamination and air quality. The second is agriculture definitely has a nitrogen problem. There's a group led by Cornell and Buckler, where we are looking at the whole nitrogen cycle in order to find interventions, create plants that can have a different growth profile, so we can circularize that nitrogen economy using improved crops. Part of these improved crops, and this is funded by FFR, is about planting earlier, so the supply of nitrogen meets better, or the demand of the crop is better tuned to the supply of nitrogen, for example. Something we are doing here at University of Florida with my grad students focus more on the nutritional aspects, whether it's strawberries with the vitamin C, for example, or Melanie with the breeding program for broccoli with increased folate and omega-3s, for example. And we collaborate with the Center of Plant Success, which essentially is looking at how to improve the predictive skill of models so we can hasten rates of genetic gain. And finally, once, let's say, we have these nutritional products out, it's not just about creating, making more available, but thinking about how we can provide access to humans and communities and to adopt these healthier foods. Broccoli is a good example because there's a kind of a trade-off between the glucosinolates and the pungy flavors with the adoption, and of course, the healthier it is, a little bit the flavors are compromise the adoption. So there's an opportunity there to use breeding to change the flavors, make it more accessible, growing in different places, for example, in Florida, to increase that availability, the access, and the adoption of these healthier foods. So could I go into talk in the next hour, 40 minutes is review a little bit of the work we did with Corteva, work that Taylor is doing in strawberries, and some of the work with the nitrogen. What this is about the now. What can we do about now? What can we deliver more with less? Essentially, reduce that nitrogen fertilizer consumption through better management, produce more carbon with less water, whether this is in strawberries or maize. I think maybe the community of ag engineers are less familiar with breeding, so let's start with a very simple description of what breeding is about and look at this equation. Essentially tells us we monitor progress, that delta G or genetic gain. It depends on our choices, that selection intensity, and the H square essentially is what fraction is heritable of that phenotypic variance. Essentially is the biodiversity, our option. So in a breeding program, we need a set of options that come from germplasm and crosses, the intensity of selection, which are our choices, and that will determine the progress. The key in here is that we don't need to be the best. We don't need to create the best absolute product. This is a marathon for the long run. We need to be better than the one running ahead of us, essentially. So we can make progress, and this is in the long term. The breeding progress essentially is looking for plants with desirable and heritable characteristics. So essentially look for biodiversity. We cross and we test. We generate that biodiversity and we ask the question, are we getting what we are selecting for? We are going to get something. And then we talk with dietitians, agronomists, MDs, and whoever is at the set of stakeholders, if what we think we were selecting for is what really is needed. In general, these breeding programs operate on testing millions of possible candidate plants, which are individual genotypes, and through a process of selection and sequential evaluation is going to get into the tens a year, for example, in maize. And there's a number of information flows that will help us develop mathematical models in order to make better selections with less resources. This is just one example of the work that Melanie Carrera is doing, where all these materials that you see on the right were consumed, some of them probably from the time of the Romans, because they are very old, ancient varieties in the collection of USDA that we don't know when they, what is the origin really, but they're presumed they're very old, and some of the newer ones. And you can see that not all of them perform so equally well in Florida. So therefore we can have diversity, we select, and we can start creating the germplasm and varieties which are more nutritious and adapted to the local conditions. In maize, the thing is a bit different. This is almost like a global scale. This is just one example from one of the Corteva research stations. There are many like this. You can see the size of the feeding trials. And the breeding program here, think about they are selecting females and males to create hybrids before they start going to test it in multiple environments. So on the left, think about they are crossing within a group, the males. On the right, they're crossing the females, they evaluate, they test, and the best candidates or the best material are contributed back to the source germplasm for further crossing and creation of product. Applying this procedure and with a dedicated breeding program, which objective was to essentially produce more with the same or less amount of water without an yield penalty, Corteva AgriScience undertook a major investment and created the Aquamax hybrids. And to do that, there was a significant investment in research methodologies, in evaluation, and developing of managed environments and whatnot. But also there was a significant effort in teaching internally the different teams. This is not an online course, but in the field, between the practitioners, teach each other how to manage that water and how to manage those experiments to expose that variation. And so the breeders have choices to identify the germplasm that they led to the improved drought tolerance. And finally is an extension component, which is significant, which is teach the farmers how to maximize the value of the seed, that genetic potential they purchase, and extract the most value out of it. So overall, there are more than 10 million acres grown a year with this type of material. And it's actually expanded to the European Union and Brazil. In 2 decades of drought breeding in one slide, I hope I can confer the message amount or summary of the technologies were developed, from imaging to kernels to satellites to characterize and identify environmental variables that really matters, to predict performance, to assays to measure roots in control environments, engineering to get the fleets of drones, to phenotype and collect data, to the use of these managed environments so we can separate drought tolerant and susceptible. But there's a very important role of mathematics, and particularly the crop growth models, as an integrator of all this information. So we can have columns and columns and columns of data, and we can drown in it, or we can use tools like these dynamical plant models, essentially differential equations, a type of a model to integrate the environment, the genomics data, and of course all the different phenotypes. And of course artificial intelligence comes into play as we can train these models. A little bit more in-depth treatment of these crop growth models. If f of t is a function of our phenotype, essentially what we see in the real world, that emerged from what we know and what we don't know, essentially these two simple functions. And Q of D is what we detected. This is our crop model. These crop models is a representation of what we really know, and this is important because it's a scientifically based model. This is not a data-driven model. This is an integration of scientific knowledge in terms of mathematical equations. And some of the parameters are known, some parameters are unknown, unestimable, and some others, of course, are going to contribute to the error. But certainly there, we have an option of estimated or measured parameters. And very important is a cognitive model, because these arrows are is a directed graph, not an, which is the different elements are causally related to each other. When we put this together and we start thinking in terms of artificial intelligence, we create a system which enables us to do some abduction, which is quite different from the current methodologies. I put this equation here as the simplest core model you can think of, but for a community of engineers, you can identify very quickly that the W variable, which is mass, is also in the and the water and the two equations. So again, we have a kind of a rabbit in the fox type of system, which can have multiple states. So within this dynamical set of equations, now we can think about parameters such as this omega, where you can use mixed models of some sort, where the X is a matrix of genetic markers, and we can predict those parameters based on genomic combinations. And of course, that could be trained using a Bayesian algorithm if you choose to do so, or something else. So now you have a very transparent mapping between the molecular markers, that is the genetic makeup of the plant in X, with the parameters that go in differential equation, and you can estimate these parameters based on the phenotype data you have with this, for example, W and theta. So this is a kind of a cleanest representation I can share in terms of what sort of prediction technologies are we using so we can hasten genetic gain. How it works, the simplest case, we take a prior distribution, let's say in parameters is radiation use efficiency. We take a sample, we estimate the marker effect as you saw the equations before. We can predict the value. We simulate for the multi-environment trials, essentially run simulation for each genotype in each environment, calculate the difference. If we like it, we keep it. If not, we toss it away. Metropolis Hastings is another way to be a little bit smarter about this. But at the bottom line, we look at the posterior distribution that can tell us how the data conforms to our theory. We ran a simulation study here. The way we choose to do that is looking at this radiation efficiency range, which is, of course, as you saw in the equation before, you increase photosynthesis or RUE, and what it's going to do is it will consume water faster. So if you have two environments with water and without lots of water, you're going to see a bifurcation at some point in time. So each point is a genotype, and you can see the branching between the two environments. So if we take any sort of machine learning model that uses genetic alone, which is the current approach, you can see that it's going to predict the same across environments. If you use something that accounts for that scientific knowledge to predict the interactions with the environment, you should be able to regenerate this function. So when you do that, essentially that's what you find. The ranges are compressed, and this is not surprising because we use linear mixed models, and of course it's going to be penalized or shrink towards the mean based on the signal to noise ratio. What is really interesting is this approach enables us to capture the behavior of the system in a smarter way. So essentially, if you have yield and you want to estimate RUE across genotypes and environments and estimate the marker effects, you can use that abduction that harness the machine learning search algorithm with the scientific base model, and you can generate hypotheses that now are testable. So when you do this, not just with this toy example as I show, but with thousands of genotypes and several years of experimentation, and this is work done with Tom Tang from Corteva AgriScience, we can see when you compare the correlation of, let's say, this statistics-driven or data-driven model alone on the x-axis, just look at that correlation between 7 and minus 0.3, or when you combine the approach as explained using these dynamical models combined with the genomic predictors, you can see that by large, most of the time, we are better off by using these knowledge-based models and abduction rather than using just inference or induction as using the data-driven model. So that's where we are at today. Essentially, we went from this sort of machine learning or statistical learning type of algorithm based on induction to something that we combine this data with the knowledge that will enable us to ask counterfactuals, what if and why it happened. So this put us in a very different situation as we want to design crops and design systems for these breeding or production systems, or design phenomic strategies or the managing environment or a cropping system for cases that perhaps the data is not existent, like in a climate change or with nutrition or process with increased nutrition that we are selecting for but doesn't exist yet, or for example, cold tolerance in C4 crops. So again, a radical shift between where we are at today with machine learning type of approaches to this based on induction to an abduction. So this is fundamental because what we can do now with these models, as I show, we developed with Corteva, is that because the crop growth model not only can be trained for the yield and the quality of the agronomics, but also we can run nitrogen balances, water balances, and carbon balances, so we can make selection decisions not only based on yield, but on the quality, but also on the emissions, on the quality, water quality, and carbon sequestration. Some of these things are totally incompatible with the time horizons that we have in breeding, but we can use simulation to have a sense or have a metric for the merit of the different genotypes on these different dimensions. So we go from, if you will, a multi-trait to a real multi-dimensional type of decision framework, so we can make selections not just on feeding calories, but nutrition and environmental quality, and of course, less greenhouse gas emissions. Just simple one example to illustrate when we run these simulations, as I show you with these 7,000 hybrids before, you take 2 of them and you can plot it against this evapotranspiration range, look at the stubble and the yield, but you have 2 hybrids, and it's clearly the separation that if a grower in Nebraska with often availability of less than 500 millimeters of water would be better off planting hybrid 2. And if there's a lot of water and we really want to do that, for whatever reason, the hybrid 1 is preferred. So if you're going to think about regenerating resources, now we have options, we have choices, and we can make informed choices based on the genetic makeup, the physiology, and the environmental conditions, and the frequency of occurrence of these conditions. So this is essentially an improving water productivity beyond what you can be attained just for the simple merit of growing aquamats for improved yields. So can we increase further? And this works with corn. Does it work with some other crops? This Taylor Sawyer, my student, asking the question. Of course, there was an interest in develop this crop model genomic selection model, but can we produce more with less in strawberries? When we started doing the literature review, she found out that photosynthesis was extremely low. So that started open a question, can we improve it? It's certainly half of a normal C3. And because the strawberry stands, they don't reach full light interception, there's a huge opportunity to increase yields by changing photosynthesis, which is not normally the case in maize or soybeans. So the objectives were, first, can we reproduce these low values? So these low values is an artifact because they are all germplasm, is our elite germplasm, but what variation is in checks, and can we do something about it? What is the magnitude of that genetic variance or phenotypic variance times H square? I showed you before, in a sense that we have choices to improve. So the first experiment is check. We ran in the lab for one of the commercial hybrid, commercial clones, and the photosynthesis was spot on what the literature review yielded. So nope, there doesn't seem to be an artifact of all germplasm, but certainly is in the modern germplasm. So the second question is the variation. Maybe this just happened to be the case, and all the checks that we have managed, they are pretty consistent. These measurements of low photosynthesis are quite repeatable. So it's not a problem of the measurement of the assay. So we went, or Taylor went, she's a very curious student and very resourceful, got the Lycor 6400s and started measuring in a breeding trial photosynthesis for several clones. And what she found out as the genetic variance is 4.17, is significantly different from zero, and is about 25% of the value we measure for the checks. So there's variation. We have a choice. It's possible to improve. We can make selections, as I show you in the plant breeding equation. When we look at all the clones that were in that breeding trial, all this, the open dots, and I shown in this figure, we can look at the plot, the checks. The checks are around that 16 to or so micromole per square meter per second. And the selections are much higher, significantly higher. And some of them are below with, or they're attained with lower transpiration. So that would tell us is there's a possibility not only to increase photosynthesis, but also increase photosynthesis and reduce the transpiration or increase intrinsic transpiration efficiency. So now we change a little bit of gears. We can, I hope I convinced that it's possible to reduce water use in agricultural systems through genetic improvement of crops. So how about reducing nitrogen loss? And here we change gears because we are going to use a little bit of the role of collective intelligence and the role of sensors, which is focused on the IoT for ag. We generated a little bit of a farm bus here with a number of agents and different levels of in degrees or connectivity between the agents or these virtual farmers. For each of these agents, we set up a system in which one of the farmers will use the fertilizer and yield response, pretty much the standard procedure that they use. And the other farmers are going to use sensors and are going to implement this adaptive system or control system within and between seasons to minimize the externalities. So essentially, each of these agents will put and share information with other agents, so they can kind of play a little bit of a game theory there. We are using the crop growth models of these dynamical system models to generate the mean yield response to nitrogen. So these agents are going to sample from there. And then we are going to look at the aggregate value for thousands, 15 million simulations, if I'm not, if I remember correctly, that shows that it's possible to reduce the nitrogen losses by 40 grams per square meter, which is a substantial amount, without losing yields, at least in any significant manner. What was kind of surprising, of course, this is not a, there are many factors that can contribute to the absolute amount, but there's a very clear pattern on the level of connectivity that the farmers have in that sort of farmer sharing network of information on the stability of the reduction in that nitrogen loss. So therefore they are not switching and the information is not get lost and the technology is discarded. So here you can see that between -200 and 200, there's quite a bit of scatter. If a farmer starts losing 200 kilograms, is going to forget about that technology, is going to say, it cost me money, is going to drop from the program or not insist. However, by sharing the information, they can assess whether it's a problem with their operation, is something systemic, or is just bad luck. So now we can iterate now, and we start working with the growers in order to bring more realism to this and start implementing and testing the technologies with the center. So that is what we can do today or in the short term. In the longer term, what is possible, and I encourage you to look at the CIRCA project led by Ed Buckler, rethinking all this nitrogen cycle that is going to take a whole talk in its own. So in the next 10 minutes, hopefully I will cover something about the systems breeding, which instead of using this for corn or broccoli or strawberries for nutrition and productivity, we can think differently in terms of how we can spur economic development by changing the patterns of the land use. So one of the problems we have is that these dual cropping systems like the safrinha in Brazil, they are possible, but they're slow to develop. If you look at in this figure, it took Brazil in the 80s till today to get to the 15 million hectares that they're growing in the Mato Grosso. But it was a 40-year journey of trial and error, from soil amendments to the genetics that they were required to plant at the right time, mature at the right time, and make a proper use of that water, limited water resources that they have in the dry season. So the story may go a little bit different because soybeans, they need to look at the second crop because they couldn't do continuous soybeans. So they went from this full season to a shorter season to avoid some of the pests in addition to that. But certainly they went from full season to something that can tolerate a little bit of flood because they need to harvest in the middle of the rainy season, to maize that it was irrigated, to something that now need to have drought tolerance because it's grown after a soybean in terminal drought condition. So this was co-evolved over time. But the question, if you look at these two graphs in terms of soybean and maize dimensions, which is just given the phenology, how many of these systems are not visible? It doesn't exist. Simply, we breed for monocultures, not for the system. The agronomists have a very limited set of options to start discovering or testing this production system because their genetics are simply restricted. So the challenge for a breeding system that will seek to improve cropping systems and not just individual crops, and that's what we do, is that this is a combinatorial problem. If we think about in any stages or small program testing 1,000 genotypes for crop 1, and we want to test another 1,000 genotypes for crop B and crop 3, you can see that very quickly this goes to an unreasonable number, which is impossible to explore empirically. So one of the opportunities we have now that we created these or use crop growth models with genomic selection is if the system, as I show you, work for maize, is working for strawberries, at least for the photosynthesis, which is part of the equation you see there. We can think about using it for broccoli to other crops. If we work for one crops, we can think we can use for multiple crops and therefore optimize not only the management of one crop, we can start optimizing the management when these crops are grown together. So essentially, this is where we challenge the paradigm, and instead of doing the breeding for monocultures, we can implement a system, breeding system that is not that different from what I show you for corn, but instead of running a breeding program for a male and the female, look at it for the, if you will, that hybrid vigor at the plant level, we are looking at, as analogy, the hybrid vigor of the system itself. So we need to breed for sorghum and broccoli, for example. We test it conditional upon each other, and we make individual selections, and we do the further testing. The key technology that will enable all this is that sort of combination of crop model with genomic selection in order to find the possible solutions in that bidimensional space that is impossible to explore empirically. So where we are at essentially is improving the crop model genomic selection. We are building models that are suited to running high performance computing. We are implementing a broccoli breeding program for improved nutrition. and certainly adapted to the Florida conditions, working with Corteva on the sorghum to see how we can demonstrate this system. So essentially, we're going to test, learn, and iterate as much as I need it, and certainly we're going to teach and share these with stakeholders and train the next generation of students. So we talk about availability very much, producing more with less, or use less nitrogen without an ill penalty. So there the question is, okay, we have this breeding program for broccoli, it has a better nutrition, they have phytonutrients. Usually broccoli is related to reduce cancer incidence or reduce risk of cancer, is even cardiovascular diseases, even autism, which is kind of meaningful to me. So we have the thing available for consumers. The question is how we're going to make accessible to consumers. And here is where we are working with the World Economic Forum and the Brain Health Initiative, and one place which essentially is this brain, body, planet wellness, taking aim at this environmental and health crisis. So the axis essentially is thinking about how we can harmonize the urban development and rural development in a way that we go from land in agriculture or dedicated to agriculture, moving to urban settings, to something that is more, which is in harmony, whether it is at the different sectors of or different social realities or socioeconomic realities. And this is where many actors come to place, from Habitat to Humanity to some of these agrihoods to circular organic farmers working in the Sarasota area. And of course, the adoption, there's an important component that is going on with education, extension, public health, and culinary and health literacy. So essentially, one of the projects which is quite exciting is how to design the systems, going from a place which is mostly houses to something that is combine or harmonize the places where we live and learn and play, why not? At the same time, we can start producing the food and eliminate. Of course, this is not the most illustrative because we can design the whole thing, but how we can eliminate essentially food deserts and develop and deliver to society healthier foods. So in terms of interest, and my project foundation essentially is a summary. I think we can reimagine agriculture solution to climate change and this human health crisis. We are working on broccoli for a reason, because we believe that. I think we need to shift the mindsets and approach the problem as predicting or stop predicting what we cannot predict, but managing the complexity. And this is elements of emergency engineering we have to play and have to bring into agriculture. And finally, I think we need to create and apply a framework to plant breeding so we can deliver on the demands of modern society. So with that, I would like to thank you for your attention. I'd love to engage in a dialogue with all of you.
Do you have any questions? Feel free to unmute if you'd like to ask anything verbally. Okay. Well, thank you so much, Charlie. We really appreciate this interesting and informative presentation. I'll go ahead and just give a plug to close out the session today. We are looking forward to exploring more CBS topics further during the upcoming CBS Day, and that's going to be July 9th in Omaha, Nebraska. CBS Day is being held in conjunction with the 2023 annual international meeting and will feature a full day of technical programming on a variety of CBS-related topics. I'll go ahead and drop a link into the chat for anyone who would like more information. Registration is now available on site in Omaha. Last call for questions. Okay. Well, thank you so much, Charlie. We appreciate your time and expertise today.
My pleasure. Thank you so much for the opportunity.
Yeah, thank you. Have a nice rest of your day.
Bye.